I'm Kyle Martin, and I'm going to talk about the role of quality assurance and quality control in monitoring. So things you should take away from this presentation are: what are quality assurance and quality control? How are QA and QC different? What does QA and QC look like for AIM? What do we mean by calibration, and why do we bother? We need quality data. The data we collect needs to be sound and trusted, allowing BLM managers to take into account when making important management decisions. Neglected, disorganized data can cause very different management decisions to be made down the line as opposed to clean, quality data. The quality of the data collection affects the analysis of that data. The results of the analysis affects the interpretation of the data, and the interpretation affects the summarization of the data for the managers. Without QAQC, we would not be able to trust our data. Quality data requires a well-defined data flow, active data management, and continuous QAQC. Everyone in the data flow needs to know what their role is for QAQC, from the crews up to the national AIM team. At each step, whoever is in control of the data needs to be actively checking the data for errors that may have been missed by the previous person in the data flow, and flagging things for the next person. Don't just move the data along. Each person in the data flow needs to have an active role in QAQC, because we need to make the correct decisions for the cattle in that photo. So what is the QA in QAQC? Quality assurance is making sure we have quality data by taking preventive measures in the collection and handling of that data. It's like having a friend proofread a paper you want to send to a journal. You still have time to make corrections. Quality control is the reactive process of checking for errors in the data that has already been collected. Sometimes we can fix those errors by going back and checking the original data sheets, but most of the time we will be keeping track of errors through the known errors form. This would be like after you sent your paper to the journal. The journal may have published your paper, but you can still give a corrections statement for any errors. There are two types of errors, sampling and non-sampling. Sampling errors happen because we don't sample every square meter of BLM land. Sampling errors are no one's fault. With good sample designs and method selection, we can reduce the sampling errors. Non-sampling errors are pretty much just human errors. These errors are just spelling something wrong or forgetting to record something. It happens. But with QA and QC, we can reduce these errors. So here are some ways that we prevent these errors. Trainings make sure we are all on the same page when using the methods and what is expected of you, for us all to collect consistent data. Calibration makes sure the training sticks and that you, the crews, are consistent as a team in your data collection. When managing data, it is easy to save over files or accidentally modifying a value while looking over data, so it is important to be careful, especially after a long day in the field. To reduce errors, always try to enter data straight into Collector in Survey 123. There are more chances to introduce errors when entering data onto a paper data sheet, and later transferring that data to Survey than just entering the data straight to Survey. But most importantly, check your data before pulling up the transect tapes and leaving the plot. The crew lead should have a checklist to go through before things are packed up. If you are still at the plot and you find some data missing, you can still collect it before leaving. Calibration is very important. We don't know what the true value on the landscape is, so calibration at least makes sure that we agree. With good methods and definitions, we should end up calibrating to something close to the true value. When calibrating, talk to each other about what you see along the line, like what gaps you see, and what you would call a specific pin drop for LPI. Calibration is required in a few instances. After training and before you survey your first plot, your crew will need to calibrate. After that, every 30 days after a calibration, you will need to calibrate again. If you move to a new ecosystem, you will need to calibrate, like if you went from sagebrush to pinyon juniper. And if you gain a new crew member, you need to calibrate again so that the new crew member is on the same page, even if that new crew member is only helping out on one plot. We calibrate on line point intercept, height, gap, and species richness. Each crew member will collect data for these methods on the same line. For LPI and gap, the indicator percentages of each crew member will be compared. The crew needs to be within 10% of each other to be calibrated on that indicator. For instance, if a crew has 14%, 26%, and 21% for bare ground, they would not be calibrated for bare ground and would need to do the entire LPI method again. For height, the crew needs to be within a count of 2 to be calibrated. Height calibration is not how tall the plant is, it's counting how many times height was measured on the line. For species richness calibration, the crew's species count needs to be within 2 counts to calibrate. Don't worry about identifying the species, it's the count of different species. Remember, 10 to 2. That's the difference threshold we are looking for. We do not calibrate soil stability or texturing, but make sure your crew knows what is expected for soil stability and practice soil texturing together. You could also ask your project lead for lab-tested soils to practice with. This foil the color figure is not quite the metric we use, but it is very similar. This is deviation from crew mean for about 100 crew members from about 40 different crews. They're only compared to the other members of their own crew, not all the data from all the crews. Some indicators are easier to calibrate than others. In the figure on the left, the bottom center and bottom right involve litter. You can see that those box plots are much more variable between the crew members than, say, the basal cover in the top center. In the figure to the right, an expert calibrated with a bunch of crew members. It's helpful to have an expert come out to the field, but if the expert has not been practicing and calibrating with the crew members, they could be an outlier that can skew any data that they personally collect. This slide is just to remind you of the workflow of setting up and conducting a project through Collector Survey 123, and that it is important to know who does what or who should be doing what, from the field crew to field office to state office to the NOC. Because if someone waits to do their role only when absolutely necessary, that is usually too late. Quality control is about quantifying errors. Checking for missing data like missing the entire line of LPI or missing a % clay in a soil pit is QC. QC can be checking to make sure that we're implementing the protocols. Calibration can also be QC when it reveals that a data collector has been systematically making the same mistakes which would compromise their data. And getting a soil sample lab tested to find the true value for the soil texture can determine if the crew collected soil textures are accurate. For QC in data management like archiving and analysis, it is common to find errors only when the data needs to be used or manipulated. These errors need to be fixed or recorded. Remember the 3 C's. Are the data complete without missing methods or observations? Are the inputs for the data correct? Make sure a 0 is not an O. Are the data consistent? You don't have a tree code entered on an only sagebrush plot, right? Metadata is data about data. An important metadata QC is the known errors form. If after you leave the plot and find errors in your data, you will upload a known errors form for each error. The known errors will live with the main AIM data and let the data users know what the status of the data is. The eval status of a plot is also metadata. Please keep the plot's eval status up to date in Collector every week during your season and keep a paper plot tracker handy for your own use. This will let project leads know how you are doing through the season and will also help future data handlers track down possible errors later. Data management refers to a bunch of stuff. This list is some of the important things for us to keep in mind. We have predetermined data structures in the form of Collector Survey 123, the metadata, and the formats we share spatial data in. Maintaining the same structures and standards means that our data are easy to use and combine over time. We also keep records of who is responsible for what and when. That helps when we want to track down problems in the data later. Remember, you can find almost everything you need to know in the data management protocol, plus new protocol documents we created for field crews and project leads or coordinators for ease of use. Check that out first if you have questions about data management. If you can't find the answers in the protocol, ask your project lead or state lead. Established data structures and rules help us keep track of our data. These rules and conventions can be found in the data management protocol. There are 6 main data types that we collect for a complete AIM data package: the field data and calibration data, the plot status and collector, the known errors form, the photos form, and the implementation summary. So QAQC means vigilant data handling, active data management in all steps of the data flow, and known roles and responsibilities. For successful collection, storage, and use of quality data, everyone in the process is responsible for quality assurance and quality control. That's it for this presentation. Thank you for listening.